right, so first I want to thank you for inviting me here, or thank you for introducing me and thank you for inviting me, because I feel strongly like the ambassador of the whole kingdom here, and that's the fungal kingdom. And as Søren said, my research is sort of in the interface between evolutionary biology and mycology. That's where I have my identity. And there has been some studies we have done over the years that touch on speciation, but I will bring up a particular case today. So I'm not going to show you X number of years that I've been working, 30 or something, about speciation, but I will give you one particular study example where I selected it because I think it is very interesting and fun, but also it might provide a sort of a model for how speciation or new species can emerge in the fungal kingdom, and also in other species where we do have a strong non-self recognition at the vegetative stage. So the project I'm going to talk about is about pleiotropy of genes who are important for non-self recognition at the vegetative stage, pleiotropy of those onto sexual incompatibility. And we have shown a case where that has been, this pleiotropy has led to what we think is the emergence of two reproductively isolated groups in sympatric population. And possibly this is a universal mechanism of speciation in fungi and also in, for example, in colonial marine invertebrates and others who readily fuse vegetatively and have a strong non-self recognition mechanism. So by the end of this talk, I hope you're all with me here. Maybe you're not already, but you will be in the end. But also, before I even start, I'm going to say a few words about fungi in general, because I think some of you guys here think fungi are weird, and they are not. Okay, fungi, they are, first of all, they are one of the most diverse groups of eukaryotes. I mean, we have 150 species, now I see if I get this one to work, who are just formally described and have a morphological description. But of course, much of their life is very cryptic and not visible for us by the naked eye. So recent estimates from DNA analysis have shown how much we have underestimated the number of species out there, to be up to even 6 million. We have a suggestion now from DNA data that there are 6 million species of fungi out there. Only a handful of those, or 150,000, are described. So you can see how much of unknown diversity there is to keep exploring. They also include, I want to give say a little point about this, that they include both micro and macroorganisms. I mean, are fungi microorganisms? Yeah, to a certain extent. For example, we have the yeast, Saccharomyces cerevisiae. We know this one very well from different studies, but also from beer and wine and bread. And, you know, maybe we don't think about it all the time, but that's the, we can thank Saccharomyces for all of this. But it's definitely a single-celled microorganism that's part of the fungal kingdom. But most of the fungi are actually multicellular, and they grow by extending their single-cell wide hyphae and form mycelial networks, like on this image here. And the biggest organism on Earth is actually one of those mycelial networks they found in Oregon, California, or Oregon in America. And it's been traced to have, you know, basically the same mycelial individual covering 8 square kilometers area. And that's formed by this Armillaria ostoye, which is a honey mushroom that's also found in Stadsskogen in Uppsala and everywhere you go. So this is the fruiting body of that one. So fruiting bodies that we pick are like they're only formed from these extended underground mycelia. Fruit bodies are like the apples of the apple tree, basically, except that we don't see the apple tree because the mycelia is usually hidden underground or in wood. The fruit bodies can also be super massive, actually, and this is the Swedish Swedish record is held by jätteröksvampen, Langermannia gigantea, shown on this one. So fruit bodies can be tiny, fruit bodies can be really big. Species can be only single-celled, and they can also be actually representing the biggest organism on Earth. So that's how they look. And then what they do, they also show a wide range of functions. I think we all know symbiosis by ectomycorrhiza, for example, is what we have all the trees in the forest outside. We do have the lichens, which is a symbiosis between fungi and algae and other fungi as well. We have fungi as important parasites of insects and of plants, and we do have fungi as the most important degrader of dead organic material. So they show a wide range in morphology, they show a wide range in function, and most of them we know are important for human health and economy. So we have quite a significant issue with fungal infections nowadays. They are much fewer in frequency than bacteria and virus infections, but they are increasing in relative frequency more than any other group of organisms. We do have problem economically at the forestry and agriculture from pathogenic fungi, but we do also have very good cheese from penicillium, for example, and we have many different possibilities of solving the world's challenges from fungi, for example, in providing us with new biofuel sources and things like this. They're also very important model systems for genetics. So at your genetics class, you probably remember the Rosetta of Assay from Neurospora crassa, where you learned how to study gene conversion and crossover first, second division. Jaron is laughing. You don't remember, but I can tell you all about that. Maybe you slept through that class, but I'll talk about that next time. All right, so I hope I have, that was sort of my detour around fungi to introduce you to this world. And also remind us that most diversity is yet to be explored. So, and for all of these species out there, there has to be, of course, a speciation event, right? So there's lots of speciation to study too. But the trait, okay, so let me see here. When I was invited to give this talk, I thought, okay, I want to actually give you something to take home with you that fungi are not so weird. They are quite similar. They have commonalities and peculiarities, but many, many commonalities with other eukaryotes. For example, they are sexual organisms. They go through meiosis and karyogamy. They alternate between the haploid, diploid. They are multicellular, most of them, and they have disjunct, some of them have disjunct distribution patterns, so they are quite good models for study diversification in nature. Of course, they have some peculiarities that are not necessarily fungal specific, but still they are predominantly haploid, have no difference in sexes, but mating types. And many of them are hermaphroditic, and others are isogamic. So it's actually provide a system where we can go in and study those traits one by one and see how they affect, for example, speciation and other eukaryotic processes of evolution. So, and I'm not going to give you an overview of speciation research in fungi, but in principle, there are speciation studies in fungi involving genetic incompatibilities, chromosomal rearrangements, hybridization, ecological specialization, and I also listed a few reviews here if you want to go and read about them. Because now I'm going to turn over to this particular part of fungal life cycle that I think is extra important and for my particular case today. And that's about fusion. Fusion is key to the life of a fungus. You see here a mycelium that's extending from the center. A spore grows and it grows outwards in all possible directions, but it's also then very frequently anastomosed or fused. You see on this picture, you see the red arrows pointing at fusion points of an extending mycelia. So they are sessile, they don't move, and they expand by hyphal extension, but then the fusion of neighboring hyphae facilitates distribution of water, nutrient, signaling molecules, and there's plenty of studies showing how advantageous fusion is. If you use a mutant in Neurospora crassa where you can actually all mutants of all genes exist, as it's a model species, you can do experiments to test how fitness get very reduced when the fungus is not able to fuse, because the fusion allows this sort of cytoplasmic mix needed for the fungus to act as one individual. They also fuse, of course, at the sexual cycle. So you have here 4 different photos. One of the small, two of them are from vegetative growth or somatic growth, if you want. The germling fuse, the hyphal fuse, the mature hyphal also fuse. So germling fuse when they are just germinating spores and they fuse, and the hyphal is more mature hyphal that branches off and fuses. When you're in the sexual cycle, of course, there is a fusion step at fertilization that's more universal to eukaryotes, and then when they form their fruiting body, they also fuse. And of course, you need, when you do this as a part of this vegetative growth, fusion at the vegetative growth and also sexual cycle, you need to express self, non-self recognition at both vegetative and sexual fusion. So if a fungus fuses with something that's not itself, it's going to be as detrimental as it is good to fuse with yourself, if you know what I mean. You're going to be exposing yourself to lots of cytoplasmic double-stranded viruses, or you're going to have, there's lots of maladaptation you can get by fusion with something that's not yourself. So fungi really express a strong non-self recognition, both at this vegetative and sexual fusion. So here, so there is evidence for pre-fusion recognition and communication. So here's a movie from the study by Andre Fleisner some years ago, where they found that they labeled these signaling proteins and found that they were oscillating between two germlings like this. And as you can see, it's oscillating so that one is talking, the other one is listening, and it's very time specific. Now they fuse, and the cytoplasm is flowing through. Okay, now we start all over. You see, one is, the labeling of the signaling molecules show how they go to the tip of one. The other one is now talking. They're quite good, actually. Say that fungi are simple, but this is a bit more advanced than sometimes when we try to have a conversation. When one is actually listening to the other, and now that one is talking. And then also, they don't only talk, they actually decide to home in, because these are genetically identical. So they are part of this sort of the same communication group, or they are genetically identical at those loci determining this mechanism. So we do have evidence for pre-fusion recognition like this. But most of the recognition of non-self typically take place after fusion. So here is 2 panels. The upper panel is not such a fun movie, but upper panel shows a successful fusion of 2 hyphae, or 2 mycelia that are identical at the recognition loci. So the arrow points at the hyphal fusion point, and you see it's not very much dramatic. It's like, okay, there's a vacuole here, but other than that, there's just cytoplasmic flow. Panel B, on the other hand, is 2 hyphae meeting that are not 100% identical at all these incompatibility loci. And after just half a minute, you point here arrows to the arrow points at 2 of the septa that immediately closes off. Now it's time to close the door. After closing the door to this cell, vacuoles start to be formed, and vacuoles burst, and after 20 minutes, this particular cell is dead. While since you close the doors, the other cells are sort of saved from this. So it's not a very costly procedure of sort of terminating this fusion event taking place by 2 genetically not identical individuals. So this is something governed by a number of loci. And here is also what it looks like in the lab. So if you have a 9-centimeter Petri dish that I think most of you have seen, and here is an example where you put 2 individuals next to each other, 2 inocula next to each other of mycelia that carries the same alleles at these interaction loci. And you see, you can hardly see any interaction zone here. They just basically form one uniform colony. But these ones are an example of 2 genetically distinct. And then what we see here as a cell death on this panel B here shows up on the, you can see it by the naked eye that provides a clearing zone where these fungi do not fuse. They clear or form a barrage. They're just not going to fuse. And this is a very specific mechanism that fungi do have. But the dogma here is that fungi fuse first and ask questions later, and without spilling too much of that energy. And this capacity of discriminate self from non-self, of course, is not fungal specific. It's necessary for all fundamental life. You have to know who am I and who are you. I mean, you sit next to each other, you don't fuse because your arms touch each other. But if but you have to still know who am I, who is a possible mate, who is a possible conspecific mate, and who am I now? Your body needs to know when you're introduced by a sort of pathogen. So you need to know, like, who am I, where do I start, and where do something else start. So this is very universal. And these there has been many different models of these non-self recognition systems leading to speciation directly or indirectly. So I'll go through a few of those examples. First, I'm going to just say that these are typically achieved by products of highly polymorphic genes. Of course, you have to be very specific to know, okay, this is you, and you know, if it's allelic interactions, these have to be really specific. They're often found under negative frequency dependent or balancing selection. Of course, if you're very common, you're not going to provide any fitness advantage. So you're going to reduce fitness, and then until you become very rare, you're going to reduce in frequency until you become very rare, and then your fitness effect goes up. So in the over time, your frequency as an allele involved in non-self recognition, you're going to fluctuate around intermediate frequencies. So this is important too. And often they display signatures of rapid evolution. So of course, this would be a case where you have an indirect effect. You have a possibility of an indirect effect where rapid evolution lead to genetic incompatibilities between diverging populations. And this has been shown, for example, in plants, that mismatch of immunity genes result in lethal autoimmune reactions. And this was proposed by Bombliss and Weigel at the time to be a common speciation mechanism in plants. So you have, as a byproduct of these autoimmune genes in different populations, you have hybrid necrosis. Suddenly you have two emerging reproductively isolated groups. There is also examples of direct effects, for example, by interaction between different forms of recognition. So if you mention this, if I just bring up this example of overlap between MHC and mate choice invertebrates. So we know that in vertebrate populations, the niche differentiation can lead to sort of different parasite exposure that leads to different MIC allele composition, and that leads to then assortative mating. So that's also proposed by in this paper as a general mechanism to accelerate speciation in mammals. So there has been different sort of examples of the role of non-self recognition and pleiotropy in speciation. So the case I'm going to bring up now is where organisms exhibit fusion and vegetative incompatibility. Pleiotropy of vegetative and sexual recognition system may lead to speciation. And here's a tree from this paper by Brusini from 2013 showing that actually the ability to fuse at the vegetative stage is not a fungal trait. It's found in all major eukaryotic groups. But the description of incompatibility reaction at that somatic stage is actually confined to the fungal kingdom and to the colonial marine invertebrates. So this sort of model applies to those who have this sort of phenomenon in place. And the people who did the job is primarily Lorena, who's here. And Lore was my PhD student, and she's actually also one of the receiver of the Crafoord scholarship. So I'm very glad to also talk about this today for that reason. Lore, where are you? There she is, yeah. And she's flanked here by Ivan and Aaron, who are two skilled postdocs who were at the lab at the time. So we did look at this in Podospora anserina, which is, it's our study system. And Lore drew here a life cycle for me to show today, where we have a horse. Horse eats grass, and grass is full of the spores from this fungus. When the spores go through the digestive tract, they sort of get prepared to end up in the dung, of course, and when they end up in the dung, they germinate. And they germinate to form mycelia, and they form this fruit body within the dung. The fruit body is not the same size as the horse, but the fruit body is much less than a millimeter high. But Lore wanted to show how it looks. So it's not drawn to scale, but within a feces, you have tons of those kind of small perithecia that each of them is less than 1 millimeter big. So that's a simplified or the necessary part of the life cycle that you need to know to understand this study. In Podospora, there are those, I have already mentioned this allelic or the genes governing non-self recognition. And these are referred to usually as het loci or het genes. And the het stands for heterokaryon incompatibility. And you have allelic variation at those het loci in Podospora determine incompatibility after hyphal fusion. So here's a schematic picture of what's going on. You have one hyphae of individual A, another hyphae of individual B. You fuse, And then if you have no differences, no allelic differences at any het locus, you fuse and you form a compatible so-called heterokaryon. If the nuclei are different, that's why it's called heterokaryon. But if there is genetic difference at a single het locus is enough for this phenomenon to kick in, that you close the septa, the fungus closes the septa, the cell between the septa closed will die, and there will be hyphal compartmentalization and cell death, while the rest of the fungus will move on as nothing happened. And it's the allelic interactions at those het loci then that determines whether you're compatible or not at this stage. And of the 9, we think this system was really suitable for this analysis, because the 9 genetically identified het loci in Podospora anserina, 6 have pleiotropic effect on sexual function. So you see here are the loci. They are called everything from het B, het Q, het C, different het combinations. And some of them are allelic, so that the interaction between 2 alleles of the same locus determine compatibility. And some systems are non-allelic, meaning that the allele of, for example, het C interacts with the allele of het E to refer to as compatibility. And the gray bars here, or the gray arrows, represents those interactions that have a pleiotropic function between vegetative and sexual compatibility. So you see, we have this case of a fungus where the self-non-self recognition is quite well established. We know which loci are involved, and we also know we have a pleiotropy on sexual function. So let's go to and see what happens if we look into natural populations. So we took advantage of the so-called Wageningen collection of Podospora anserina, and for those who don't know it, Wageningen is in the Netherlands. And here is a photo of the Wageningen campus. This is our study field. It's not a beautiful seashore in Queensland, but it's quite convenient. You go to this grassland, you find the dung from a herbivore, and in this case it's probably a small rabbit. And then you bring this into the lab, you put it into the right humid conditions, and it will start fruiting. And these, again, are not the same size as the dung. These are very small, but they are enlarged here. These fruiting bodies will shoot their spores, and then you can isolate them to have cultures collected from different perithecia or fruiting bodies from the different dung samples. So we had a collection with a nice contribution of our collaborator in Wageningen of 106 such strains, spanning 20 years of sampling in this time interval around Wageningen. And then we had some other strains as well in our. We just sequenced them all with Illumina and PacBio and everything we could do. And they are, as you remember, well, the fungal genomes are not so big, so we could easily get good assemblies, telomere to telomere assemblies from these to be able to compare them to each other. In order to study speciation, of course, we need, or our model really inferred that we need to know that this fungus is actually doing outcrossing. And we have been working quite a lot on spore killing on meiotic drive, and we found that meiotic drive happens. This was actually a qualitative measurement of outcrossing taking place. We also looked at LD then from in our population, and by chromosomes it's 7 chromosomes, and we see that the LD reaches about 0.2 at distance below 3.5 kb, and what this means is how much recombination you have. And it's this sort of decay curve suggests there is, it's similar to what's been found in other fungi that are sort of partly selfing and partly outcrossing. So what we did was genome scans for diversity and Tajima-D to look for signatures of balancing selection. So here will be 2 examples, chromosome 1 and chromosome 3. On the upper panel you have diversity, and you see it's very low except at some particular peaks. And those peaks sometimes coincide also with a peak of positive Tajima-D, suggesting sort of population subdivision or equal frequencies of alleles. And these also coincides with a locus, for example, of, in this case, now I have to see, you see better than I do, one of the het loci, and in chromosome 3, you had also sort of the peaks corresponding to different het loci. So we kind of confirmed too that heterokaryon incompatibility is at place, and it's functioning as this fungus grows there in Wageningen. We could also do plots of these allele frequencies of the known het genes over time. So we didn't have really large sample from each year time point, but we could still see that they sort of obey to this typical pattern of intermediate frequency, selection for intermediate frequency. So our different alleles at het Z, het C, and het S shows intermediate frequency over time. So we have a population that's outcrossing, it has somatic or vegetative incompatibility, and the het genes show the typical signature of balancing selection. What we also found was within this population that we thought, well, we had the suspicion that we had two different sort of mating groups here, and this would be typical for fungi because we see nothing there. This is basically a DNA that shows us that these, DNA shows us, but also in this case, we can bring them in and cross and look for a score for fertility. And see that when we bring them into the lab and score them for mating success, we have two intermixing groups that do not seem to mix very well. They don't mate very well. And these two mating groups now highlighted with green and purple fit with the whole genome SNP data as well. So the points here represent all our genomes we have, and the colored ones represent those for which we also have mating data. So the gray ones we don't have mating data from. So we see that these reproductively isolated groups are supported by SNP data. And when we look chromosome by chromosome, it seems like this differentiation is really boiling down to chromosome 2 and chromosome 5 in particular, while the other ones are sort of intermixed. So whatever is found on chromosome 2 and 5 must be something that governs or leads to this reproductive isolation. So we took and separated the pools of strains belonging to each of those reproductively isolated groups and did an FST analysis. And again, we could then zoom in on, we could again see that it's peaks on chromosome 2 and 5, but then we could also zoom in where on the chromosome 2 and 5. And again, we had a possible location of the het R and het V loci on these particular chromosomes. So we are starting to narrow down the two loci important for this reproductive isolation. This was an interesting case because this is one of the known non-allelic interactions, and they were also one of them was not known. So now we have to go back to see, what we have to back to do the crosses and see exactly which combinations had fertility and which ones had infertility. And this is how it looks approximately. So you cannot see it probably, but there are small fruiting bodies here in an RV. Here you have a cross between two genetically identical strains, and they are then very fertile. They are identical at that incompatibility locus. As same goes here, but if you cross this genotype to that genotype, you have no fruiting bodies formed. So we took one step at a time to confirm the function of these genes. The genetic basis of this locus was not known, so it was known since a long time ago, but we didn't know which gene was involved, so we had to do positional and complementation cloning together with Sven Saupe at the University of Bordeaux, where we zoomed in from these FSD plots to find what are the actual gene involved in here, because here would be the predicted annotation of the genes in that part of the chromosome, and then we could home in to what gene was actually important. And then we could go all the way and confirm that by site-directed mutants that there's this domain PAMT1 confirms a methyltransferase activity. So now we know the function as well. And of course, we did all the, according to the book, of deleting and bringing back to the genomic background to say this is not only deleting the phenotype, it's also bringing it back when we bring it into the genome again. So we have to go back to the nature. We see there's two genotypes that define these reproductively isolated groups in nature. But as this is a two locus interaction, we would expect to see all four possible combinations. Where are the others? So one of them we found was actually lethal, so that's easy. But then this genotype is it's very low frequency all the time. And if it would be random mating, you would see this one as equal as those two. So we still weren't really sure, do we see two reproductively isolated groups governed by this genotype, or is it something else going on? So we then thought we had to go back and see, is there anything we can find in there, in the field, that would suggest ecological isolation, for example, different herbivore dung. So we looked into the horse dung and the rabbit dung and the sheep dung, and see that we found the two genotypes in equal frequency in all dung. And then we thought, yeah, but it could be a time problem here. But then we actually looked at the dung samples one by one and saw that in cases where we had in the same dung, we actually had both taxa. So it's really not an ecological isolation. They are there at the same time, at the same place, but they just don't mate. So then we had to do some simulations to explain under what condition can these two genotypes actually drive speciation. Is there anything we can see in the life cycle that we can elaborate with? So first of all, we have the frequency-dependent selection that drive up frequency of a rare allele. That would actually be a very strong component of this prediction. The other one, the other important parameter is rate of selfing, because how much you outcross or self will influence how much you link those two together. Of course, there is, as I showed you before, it was a quantitative or sliding degree of sexual incompatibility in those interactions. So we had to put that as well into the model. And then the easy case, that combination was lethal. We also put it into the model. And then we see under which conditions can we find a stable coexistence of those two genotypes in the population, and thereby support that we have this gene combination driving speciation in this fungus. So here's the simulation result. And now I think there might be a little bit, something is weird. That it didn't look weird on my computer, but maybe we can just ignore that. So what we have here is here's basically the reproductive isolation in three different levels. We had three different panels showing three different levels of selfing. And then we had also different degrees of balancing selection. And you see that if we had a selfing rate of 90%, nothing else matters. Then we have the stable coexistence of those two genotypes and the very low frequency of the recombinant genotype. So what we can conclude from this is our model holds if under a high level of selfing in this species, which we know we probably have. So that's it. Did you follow? The conclusions: fungi is an extremely diverse group of organisms. So much left to study, so many speciation events that we still have to go and look what has been driving that. What we think is that we could confirm, or we feel confident in confirming, pleiotropy between vegetative and sexual incompatibility that can cause speciation. And in our particular case, we needed a high level of selfing to maintain the reproductively isolated groups in Podosporan serina. And possibly then this sort of pleiotropy could be a common mode of speciation in fungi and also in other systems where you have this strong vegetative non-self recognition taking place. Yes, and with that, I want to thank my group. We're just outside here in the spring trying to establish ourselves here in Stockholm, and we are very happy to do that, as you can see. I want to thank in particular Lore, because she's a superstar and she did all this. And then KVA, who are my employer right now, and Stockholm University for hosting me over at DEEP, and of course, to funding agencies. And for you, for your attention. Thank you.